Hello friends, Ace Botane, next episode. This episode deals with YTQ series 16B, molecular basis of inheritance from DNA replication to transcription, up to transcription. So we can enter into the part. So I will share the screen because you have to observe the question. Then we can proceed. I think a screen came, so we can proceed. So YTQ series 16B, molecular basis of inheritance from DNA replication to transcription. So first question, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This statement is connected with. This is a statement from NCERT book, page number 88. And there we can find after the statement, two scientists' name given, Watson and Crick in 1953. So option wise, when we take option is 3. So Watson and Crick 1953. So first question answer is 3. Below it, we can find a statement that is connected with the last episode. A typical nucleosome contains 200 base pairs of DNA helix. NCERT page number 83. So nucleosome, 200 base pairs. Now we can proceed to question number 2. See, in semi-conservative mechanism of DNA, after completion of replication, each DNA molecule would have. So what will be the combination of the each DNA molecule form? So we can find, in such case, one parental and one new strand. That is semi-conservative mechanism. So one parental and one new strand. That is option is 2. And this mechanism, that is the reason why it is called semi-conservative mode of DNA replication. I think idea clear. We can proceed to third question. Semi-conservative mode of replication can be seen in. It is seen in Escherichia coli, plants, and human cells. So all are possible. So option is 4. All NCERT page number is given. This will be easy for you to locate. Now coming into fourth one. Fourth question. In Messelson and Stahl 1958 experiment, in connection with the semi-conservative mode of the replication, the process involved or processes involved, materials needed, etc. So totality of the commonness of the experiment. So first is N14 heavy nitrogen. N14 heavy nitrogen. N14 is there, but it is not heavy. Second is E. coli. E. coli is needed. Third is cesium chloride density gradient analysis. It is required. N15 light nitrogen. N15 is not light. Then fifth one, N15 heavy nitrogen. That is required. And N14 light nitrogen also required. So what we can find, second, third, fifth, and sixth are correct or requirements. So option wise, 2, 3, 5, 6, it is 1. I think idea clear. Now we can proceed to next one. That is fifth question. So when we come to fifth question, generation time for E. coli under optimal condition. Optimal condition is approximately. NCERT experiment, Messelson experiment, when we check, we can find it is given as 20 minutes. So answer is 3. And this time can be reduced when we give best rich nutrient condition. So time will be lesser. So normally it is 20 minutes. So fifth one, it is 3. Sixth one. When we proceed to sixth question regarding Messelson and Stahl's experiment, find the correct statement. N15 is a radioactive isotope, can be separated from N14 based on densities. Now, second, N15 is a non-radioactive isotope, yes, and can be separated N14 based on density. So it should be option 2. N14 is radioactive isotope, no. N14 is non-radioactive, no. So answer is 2, direct from NCERT page number 89. Now, seventh question. When we come to seventh question, it is famous Messelson and Stahl's experiment. here A, B, C respectively stands for A, B, C. So we know N15 will be at the bottom, heavy. N14 will be intermediate. And N14 will be at the top. So in such a way when we take A is N15, okay. B is intermediate N14, okay. C, there are 2 marks. So first is starting with the C, N14, this is right. Then next is N14, this is also right. So we can find, finally answer is 4. And direction given here, gravitational force. So N15 at the bottom, and 14 at the top side, and 14 and 15 intermediate. So 7th question option is 4. Now when we come to 8th one, the diagram, NCERT diagram I given here, so it will be easy for you to refer. 8th one, in living cells such as E. coli, the process of replication requires enzyme. The main polymerizing enzyme is, it is nothing but DNA-dependent DNA polymerase. So option is 1. So 8th question, option is 1. Now we can proceed to 9th one. When DNA polymerase is supposed to be highly efficient, why DNA polymerase is supposed to be highly efficient in nature? For efficiency, different criteria are there. So it is able to polymerize 2000 base pair per second. That is right. High degree of accuracy, it is a must, it is needed, it is there. It is very fast in operation, right? And totality answer is 4. Below it, you can find some explanation that also try to read. It is for convenience, otherwise time will not be available. So 9th one, option 4. E. coli completes the process of replication in 18 to 20 minutes. NCERT says 80 minutes. Page number 90. It is generally accepted. So process replication, 18 to 20 minutes. It is generally accepted that E. coli replication of chromosome takes approximately 38 minutes. So 20 and 30, that's how 2 values comes. With the assumption DNA polymerization rate is approximately 2000 base pair per second. And for these conditions can be applied. Accordingly, it can change. So 9th one, option is 4. That is why that, say here 18, 20 and other side 38, etc. comes here. This is what the reason. One is process replication, other is replication of chromosome. Okay, now coming into 10th one. When we proceed with the 10th question, how many statements are correct? Energetical replication is least expensive. No. Deoxyribonucleoside triphosphate can act as a substrate, right? Deoxyribonucleoside triphosphate can provide energy, right? In deoxyribonucleoside triphosphate, the 2 terminal phosphates are high energy phosphates like ATP, but what given here it is unlike. So what we can select is only 2nd and 3rd are correct. So option wise it is 3. I think I declare bottom I explained it, or what I told I explained it. Now coming to 11th, replication starts from a point of DNA called origin of replication, or is in E. coli. Prokaryotes have single origin of replication, eukaryotes have numerous origin of replication. So 11th one, option is 1. Single and numerous. Now below you can find some statements. See what is importance of ORI, why it is formed all this. ORI provides the starting point for process. If no ORI means DNA replication will be difficult and even will be lost. Similarly, DNA polymerase cannot do its function. In cloning vectors, again, the ORI ensures vector and its inserted DNA are replicated, helping in production of multiple copies of the desired DNA sequence. So 11th one, option is 1. Then 12. In the activation of deoxyribonucleotide, you can find 1, 2, 3 marked there. So 1 is H3PO4, 2 is phosphorylase, 3rd is energy. So totality, all 3 when we take, option is 2. All direct NCERT, page number 90. Now 30. Unwinding of DNA is helped by the enzyme. Unwinding. It is helped by the enzyme helicases. So that is right. So for 13, option is 1. Again, some explanation given. For long DNA molecules, since the 2 strands of DNA cannot be separated the entire length, due to very high energy requirement. The replication occurs within a small opening of the DNA helix referred to as replication 4. Helicases is ATP-dependent enzyme. So for 13, option is 1. Now coming into 14. During unwinding of DNA, tension will be created, which is nullified by the enzyme. So nullified by the enzyme, topoisomerase. Option is 4. And DNA gyrase, that is another enzyme. Gyrase in prokaryote can also do topoisomerase activity. So reducing tension while unwinding. So option is 4. Now coming into 15. 15, during replication of DNA, it has to unwind. Then stability of the strand has to be maintained. It is done by, because it becomes single strand, so it is done by SSBP, will form a single-stranded binding protein. Another name is the helix destabilizing protein that help in DNA replication efficiently. So option wise, 15 is 3. Now coming into 16. E. coli DNA polymerase is, in E. coli DNA polymerase dash, that is 3, is the true replicase. It is incapable of initiating DNA synthesis. It can add, but it cannot initiate. So for 16, option is 2. Now coming into 17. In DNA replication, what is the role of primase? It's a continuation like the previous question. Primase is concerned with the production of primer strand, and primer strand, that means it is RNA. So RNA can initiate, on which DNA will be added. So for 17, option is 3. Now coming into 18. In DNA replication, RNA primer is removed by the enzyme DNA polymerase 1. So option is 1. Now coming into 19. When we look into 19th question, we can find there will be a confusion. Certain question paper or question will be like this. People will be confused, purposefully doing it. DNA replication will be continuous on one template strand with the polarity. Polarity will be 3' to 5'. DNA replication is discontinuous in short segments, that is called Okazaki fragments on other template strand with the polarity. So first is 3' to 5'. So second will be 5' to 3'. So option is 2. There are certain technical terms. Leading daughter strand is continuously formed. Lagging daughter strand is discontinuously formed, connected with the term Okazaki fragments. So 19, it is 2. Now coming into 20. Okazaki fragments are later joined by. So joining enzyme, we know it is ligase. So first option, ligase is there, but prefix is different, is RNA. So not possible. So option is 4. So check each word. Okay, just like reading NCERT line by line, through the line and between lines. Now coming into 21. When we proceed to 21, DNA replication takes place in that stage of cell cycle. Cell cycle question. Here also question. It is concerned with S space, that is 3. Now 22. As we know, the bacteria is a prokaryote. In bacterial DNA synthesis, which enzyme is odd one? Which enzyme is odd one? So we can find DNA-dependent RNA polymerase, it is there. RNA-dependent DNA polymerase, no. DNA gyrase will be there, dependent DNA polymerase is there. So what we can select is RNA-dependent DNA polymerase. RNA-dependent DNA polymerase means it is connected with the procedure reverse transcription. So it is in viruses. So your possibility is option 2. So RNA-dependent DNA polymerase means it is reverse transcription. It is seen in viruses. So 22, it is 2. Now coming into 23. Typical Michelson and Stoll experiment. That is solving problem type, that way. Now when we come to 23. In hybrid DNA, if hybrid DNA is allowed to replicate for one generation in a medium containing N15 and for second generation in a medium containing N14, then what is the proportion of percentage of heavy DNA, hybrid DNA, light DNA? That's a good question, but it is direct question. So we can just expand this diagram. First generation, what happens? The first generation, we can find N50 medium. Anyway, this strand will separate. Now, for this N15 hybrid DNA containing N for a second time. So first is in the medium containing N15. So here we can find one N15 will be added. So here that N14 will be separated from N15. So again here we can find N15 medium. So they'll be like this. Okay. Now again put in N14 medium. So we can find all that we can draw separately. For all this, we can find one white will be added because medium is now N14. Okay. Now this N14 will be here, into which another N14 will be added. So altogether we can find the same order. Percentage of heavy DNA. Heavy DNA will be 0. 2 strands will not be heavy in this experiment. Hybrid DNA 3 are there. That means 75%. Light DNA 1, that means 25%. So answer is 1. Simple question, beautiful question. We can anticipate for NEET exam. And this data is based on Messelson and Stahl's experiment 1958 connected with a semi-conservative mode of replication. Option is 1 for 23. Now coming into 24. Initially, different models were proposed for DNA replication. After experiments, it was proved that DNA replication is another beautiful question. So what we can select is, it is semi-conservative and discontinuous. So option is 4. Semi-conservative. The diagram you can check. Semi-conservative. So one strand is old, one strand is new. And discontinuous. Here discontinuous, here continuous. So totality option is 4. Now coming into 25. When we proceed with the question number 25, normal Watson-Crick DNA is double-stranded. During dereplication, in certain area unwinding is needed. Means hydrogen bonds has to be broken in those areas. The enzyme involved is. Enzyme involved is not ligases. Ligases will be uniting. So that is not possible. So it can be unwindings. Okay. It is otherwise called helicases. So totality, more than one option correct. Option is 4. So I think it is able to follow. If you like this episode, subscribe Ace Botane. The questions, everything will be coming in Ace Botane channel. So please subscribe, then share, like it, comment on it. We can meet in the next episode. Meanwhile, remember, short questions will be daily coming in the night time with a quiz-like question. He will be coming within seconds also. So I will be getting it for you. If you subscribe, then theory part will be also continuing. So for time being, I think we can wind up. All the best. Take care. Bye. Thank you.